So the title of my talk is Beyond Macrocycles, and I want to talk about bond disconnections that are enabled by olefin metathesis. But before I get into that, I want to start with just a kind of a way to clear the air. We've heard this topic before, of course, the price of catalysts, and also the IP associated with certain catalysts. There's certainly when there's questions about IP, I think it's really a twofold issue. The first one is what's it going to cost, and maybe it will cost more than a non-proprietary catalyst. But I think probably the bigger sticking point, especially for those in process development who need to be able to project costs, is really this question mark of, well, we don't know what else will be included with that. Is there an extra license fee on top of that, so on and so forth. So I want to start the whole conversation by saying that Umicore's business model for the Grubbs catalyst and actually every product in our catalog is that the IP is included in the price of the catalyst. So you buy the catalyst, you're free to do with it what you want. You have freedom to operate. We don't want to stand in your way. As Phil said, Umicore acquired Materia's Grubbs Catalyst business in 2018, and so we've now unified both the IP around, and actually more recently we've also kind of consolidated with Evonik as well. So all the IP is now under one roof, and also all the catalyst structures are now under one roof. So we hope that this removes some of the questions that had been maybe lingering about olefin metathesis technology. So just to give a little overview of the different type of catalysts that we can now offer, it includes still the first generation catalyst, including those developed by Nolan with the phenylenedinyl alkene, the ones developed in the Grubbs group originally, so like the original Grubbs 1 catalyst. Although I think, Bob, is that right? Greg made this one first. Greg Fu made that one first. That was actually the first one. Anyway. And as well as the second generation catalyst, both the Grubbs type that have the phosphine ligand on the bottom, as well as the Hoveda type that have this chelating alkylidine. And actually these are now added to, basically it's the sum, the new portfolio is really the best of both worlds in terms of what Umicore already had licensed in, variations of these catalysts, as well as the catalyst that Materia had originally had access to through Caltech. And as I said, royalty-free, license included. Okay. The other thing that we've done, that Umicore has done after the acquisition of Materia, is we've tried to continue, and I think we've been successful so far, the technology collaboration that existed between the Grubbs Group and Materia. And this is something that we plan to continue going forward. And so we certainly have plans. We have, made commercialization of the Z-selective catalyst possible. We've also done quite a lot of work with the so-called CAT catalysts. These are the Grubbs catalysts that have these Bertrand-type cyclic aminocarbene ligands on the top, replacing the kind of more traditional carbene. And then most recently, and I'll leave this, I'm sure this is going to be, you'll hear lots more about these this afternoon in Bob's talk, but the latest family of catalysts are these dithiolate catalysts, which are actually stereoretentive catalysts. Okay. All right, so if I pull back to the kind of 50,000-foot view of how olefin metathesis has been used, actually I would need to go even further back to really before these predefined catalysts were developed. But at least what I've shown here are some of the applications of the homogeneous, well-defined ruthenium-based catalyst. They extend from kind of very similar to the kind of roots of the original metathesis applications, which are in the ring-opening metathesis polymerization space. So here, Materia's done a lot of work. They've commercialized these chloralkali cell top covers, which are basically about the size of a hot tub just turned upside down. They're used in chlorine production. They're incredibly tough, resistant to very harsh conditions. Another property of these materials is that they're nice and insulating, and so the Materia's also been able to commercialize these applications of ring opening metathesis polymerization to coat pipeline that'll be put actually in the Gulf of Mexico, I think around sometime this time. So anyways, the applications of ring opening metathesis polymerization, they do continue, and that's actually what Materia has now continued to do. So they've kind of broken off their activities where we're making the catalyst. They have now all been transferred to us, but however, that work still continues in new markets, and we are their key supplier. In the cross-metathesis space, I would say and this is, all the talks, many of the talks so far have been focused in the pharmaceutical field. I would say cross-metathesis in pharmaceuticals has been used kind of sparingly, but it's found a lot of applications in specialty applications. For instance, this is a polybutadiene nitrile, high nitrile polybutadiene rubber that's used in automotive belts. And it's used in a step to equilibrate the monomers before the polymerization is carried out, and it gives a much better performance of the material. There's another very large-scale reaction or application of olefin metathesis, and that's being carried out by Elevance Renewable Sciences, excuse me. Here they have in Gresik, Indonesia, a gigantic biorefinery that's doing either ethanolysis or butanolysis, basically clipping long-chain fatty acids that are derived from natural oils to give value-added products that are used as surfactants and other things. And then the ring-closing metathesis is kind of the last member of this family, we may say a major member of this family. And here, now you start to see lots and lots of applications in pharma. You also see applications in flavors and fragrances as well. And I've highlighted one specific example here where RCM is really an enabling technology that's allowed kind of a bit of a new modality, the staple peptide, to be developed. And there's actually a number of companies in this area that are actually in this geographical area that are looking at staple peptides as potential therapeutics, especially in cancer. So I think for some of the reasons I alluded to before, including maybe the questions about IP and scalability and all these things that certainly process chemists would need to take into consideration when they're looking to develop a route. I think the combination of all those factors have really led RCM to be used quite sparingly in pharmaceutical, at least in development synthesis. So there certainly are some notable exceptions, and macrocycles are one of them, right? So macrocycles are already in their own right very challenging molecules to make, as again Seb alluded to in his talk. Whichever method you're using, whether it's ring-closing metathesis or some other method. And ring-closing metathesis is indeed an enabling technology for closing macrocycles. The first clinical candidate that was synthesized by RCM, it was a macrocyclic hepatitis C protease inhibitor that was developed at Boehringer Ingelheim in the early 2000s. And I'd say to the credit of the community, they published essentially, a very long list of papers. I don't even know if I got them all in this slide, which the rest of the community has been able to benefit from ever since, being able to look at these and kind of take away the lessons that BI learned in the development, kind of being the pioneers and the trailblazers here. Ultimately, this molecule was discontinued in phase 2, so it was not brought to market. But a very similar molecule was eventually brought to commercial scale by Janssen. A very similar structural motif, you can tell. This was approved in 2014 for the treatment of hepatitis C infections. And Janssen has also published several papers on this topic, and there's also, of course, patents describing it as well. In a very recent paper, and actually I bet I should probably replace this DOI at this point. I think it has a citation now. But they did actually come out and say, yes, a ring-closing metathesis using M2 was what was used on commercial scale in this step. And of course, there's a number of other molecules that have been approved in this space, also hepatitis C, macrocycles, and again, in these cases, it's not so clear, it hasn't been published, but it's in patents at least described as a potential route to make these macrocycles. In that time period, I'd say from the early 2000s until, let's say, a couple years ago, there's a couple of outliers that have got to development scale that were not macrocycles, and this is one of them. This is a cathepsin K inhibitor that was developed at GSK, also ultimately discontinued in phase 2. But if you look at this molecule, it's not quite so obvious. Maybe you can see this long carbon chain and think, okay, I guess you could make do an RCM there. But there's plenty of other places you might think about disconnecting this 7-membered ring. And they did look at, of course, multiple routes, and ultimately the RCM is what was used on 80 kilo scale, eventually 80 kilo scale, which shown here is the 7 kilo scale, to deliver the candidate. And if you look at these conditions, when you take away some of the complexity, and actually I would say the other really important part is you take the macrocyclization out of the picture, so you're no longer needing to do very high dilution to overcome the entropic barrier. Closing a 7-member ring, you can now run at very reasonable concentration. I think you were saying, Seb, 20 volumes. You guys were having to do 30 to 50 volumes, and that's not ideal. 5 volumes is pretty good for a 7-member ring closure. And also, the catalyst loading is really not high at all, 0.25 mol% for this step. Now, I'm minimizing lots and lots of work like everybody does in these presentations. I'm minimizing a lot of work that's described in these papers. It actually did take a lot of effort in order to understand all the impurities going into that step that allowed them to get this low loading, but it's certainly something that can be overcome. The next step is a heterogeneous hydrogenation, and actually at this stage, well, first of all, the residual ruthenium at this stage is already pretty low because it's already starting at very low loading. But on top of that, it's followed by a heterogeneous hydrogenation, which is known to convert the ruthenium chloride to a ruthenium hydride. It seems as though that absorbs quite readily onto the activated carbon. I have another example from Merck that'll show that. And then 3 more steps, and they get to their desired API, and at this point, it's undetectable ruthenium. And again, in terms of how, where is that ruthenium removal coming from, there's a really nice example from Merck, again, another very hepatitis C protease inhibitor, where they make a 20-membered ring from this substrate, again, very low loading, also for macrocyclization, very nice reactor throughput for this, only 13.5 volumes. And they're adding dichloroquinone in order to suppress the olefin migration, which I'll come back to later. And then in the subsequent step, there's a hydrogenation and a crystallization. Of course, no chromatography. This was intended for a larger scale. The yield is good, and both the ruthenium and the palladium levels are already under the ICH limits, and this is not even the API. But I think there's a lot of cases where the RCM bond disconnections are not going to really jump out at you, and I think the azepane is one of them. Maybe you could work it out eventually. Spirocyclic systems are also, again, Seb talked about the ADME properties of macrocycles, and there's certainly some evidence that making macrocycles have somewhat of a privileged structure in terms of a nice balance between pre-organization and membrane permeability. Spirocycles also seem, not that I've gotten that rational explanation as to why, but spirocycles also seem to be somewhat of a privileged moiety. They're certainly appearing more and more often. And in these cases, it's not so clear where you might put the RCM disconnection. In this case, I think from the get-go. And it turns out that actually, forming this simple 5-membered ring, this spirocycle pentene, is actually very facile, tracing back to this oxyzole starting material. You can just do a double allylation at that acidic methylene, and then close the ring using RCM. And of course, the alkene, although it's inert to a lot of types of chemistry, can be elaborated pretty easily and readily to a number of different types of chemical motifs, including through things like dihydroxylation, aziridination, and so on. Anhydrous cycles can again remain to be a very common motif in pharmaceutically relevant molecules. And again, following the trend of beyond escape from flat land, beyond rule of 5, and so on and so forth, you're seeing lots and lots more saturated anhydrous cycles as opposed to the kind of more classical heterocyclic chemistry. And for pyridines, for instance, ring closing metathesis gives you a nice way to install functionality at the 3 position, which I think is a contrast to a lot of the chemistry that's out there, including the new stuff, the cross-electrophile coupling from Abidol, for instance. Oftentimes you're still functionalizing at the 2 position. So in order to install a functionality at the 3 position, ring closing metathesis allows you to trace these back to a homoallylic amine on one side, which I'll show, and either an acid chloride or an allyl chloride, depending on whether you want to get to the lactam or the fully saturated piperidine. To make a 5-membered nitrogen heterocycle, you now trace this back to, instead of the homoallylic amine, to the allylic amine. And if you look at these, I've indicated these all as stereocenters. They certainly could be stereocenters. At least control of that stereocenter is pretty well known by things like Elman sulfonimine chemistry. So this is a pretty flexible way to make a number of different ring structures, including with chiral centers. I also should note there's a really nice review from Karina Schindler earlier this year in Synthesis that's all about making nitrogen heterocycles by metathesis reactions. And a couple of examples of these in the forward sense. We've got, in this case, again, a homoallylic amine and acid chloride. In this case, they actually is an alpha fluorine substituent here, which is compatible with the reaction, which is compatible with the catalyst. And this is carried out. These are still a kind of discovery type scale reactions. I would say 2 mol% is still fairly high loading, at least from our point of view. So here you make the 6-membered ring from the homoallylic amine, or the 5-membered ring from the allylic amine. Cyclohexynones are another one, and you know, I don't think anybody's that going to be that excited about making a corinone, a fungal natural product. But I've shown this one. It has a fair bit of complexity, maybe not a lot of functional groups. Again, there's lots of different ways you could think about making this molecule, and the way that this academic group decided to pursue it is through a ring-closing metathesis that would trace them back to a chiral starting material that would be derived from pulegone. And again, it's an allylic group on one side and then a vinylation of the corresponding aldehyde on the other. This one is closed, happens to be closed as the alcohol. This can also be closed actually as the enone. And I think this intermediate in the middle might be a little bit more interesting. Again, in my past couple of years talking to customers about different molecules they might be interested in making, and in more than one occasion I've heard an anecdote about an 8 carbon intermediate that looks so simple. It's a cyclohexynone. It looks like not that complicated. Somebody's got to sell it, but then it turns out nobody sells it. And then you look at, I'm sorry, all these guys in the front row are nodding their heads, so you guys aren't just being nice to me, are you? So anyways, some of these seemingly simple molecules can be deceptively difficult to make. And I think, and I'm not going to stand up here and tell you that RCM is going to solve all your problems, but I think that it's worthwhile looking at approaches like these. And again, in light of the fact that, you know, RCM or Grubbs catalyst shouldn't really be like a forbidden fruit anymore, I hope. One area where olefin metathesis does struggle, though, is in chemoselectivity of olefins, right? So these catalysts are active on unactivated olefins. So it's sometimes hard to distinguish one olefin from another. So making substrates like this one that has three different olefin positions, you know, again, it'd be great if you could just do a cross metathesis at that position. That'd be nice. But then you'd have all sorts of product distribution of the 1,1 disubstituted as well. You also might get, you know, all sorts of rearrangements and so on and so forth. So this is maybe not apparently at first glance a very good candidate for a metathesis reaction. Probably more logical would be to disconnect along that single bond, carbon-carbon single bond, through a Suzuki-Miura coupling. And actually, to set up the Suzuki-Miura coupling is where the cross-metathesis reaction might come into play. So these catalysts are quite tolerant of a number of different subfunctional groups, including boronic esters, even right there at the reactive center. So of course, if the catalyst were to initiate onto here, you have the ruthenium right there. That's the alkylidine next to the boron. This is catalytically active. It's not so stable. It gets trapped out like a Fischer carbene. In the forward sense, again, the first step is pretty low loading, 2%, and followed by the kind of melander prep to make the trifluoroborate, and that's a 70% yield overall. So, and my next slide is actually, this is the oldest, 1998. This is the oldest citation in my whole deck. This has been known for that long, over 20 years, that you can do ring closing metathesis reaction of vinyl boronates to make, again, very saturated, including heterocyclic Suzuki partners. And again, this is something that, sure, I would grant you that maybe 5 to 10 loading in benzene is not going to be the best thing to scale, but these things can be overcome. And we have, since 1998, there's been, the Grubbs group and other groups have developed plenty of other catalysts to try. So another area, and I talked a little bit before about N heterocycles. I think an area where RCM often gets overlooked, again, because people usually associate it with very complex molecules or think about total synthesis, the examples they've seen in academia. I think for simpler molecules, it's certainly, there are opportunities to use it more often. And this is one example. N-bok-pyrroline, which, you know, the route to make N-bok-pyrroline by olefin metathesis reaction has also been in the literature for a long time. The Grubbs group published an Org Syn prep back in 2003. This is actually already pretty low loading of the first generation catalyst. It's run in DCM, which is fine for an academic prep, but again, there's plenty of other solvents you might want to try. The yield is nice. It's a 90 to 94% yield, and you know, this is a very versatile intermediate. There's a lot of different things you can elaborate it to. Of course, you can deprotect the Bok and do an amidation or reductive amination with it. But then, of course, as I said before, the double bond can be elaborated to a number of different, you know, more complex motifs like cyclopropanes. I already talked about dihydroxylation. It's also a nice, you know, I love dipolar cycle additions, and it's also there. That's a great way to do SAR exploration. Has been for a long time. Making a lot of analogs. It's a good way to do it. So Looking at these conditions, of course, this was published in the early 2000s, first generation catalyst, again, with the benefit of all the new catalysts that have come out of the Grubbs group and others, Materia decided it was worth another look. So they collaborated with Scott Virgil at Caltech, and they did a 96-well screen to look at trying to lower the catalyst loading, also get away from DCM, also improve the concentration if they could. And these are just 3 selections. There actually had also a number of substrates. This was kind of a combination of conditions and a number of substrates as well. I've pulled out the just 3 results from that screen, making NBOC pyrroline using now 0.05 mole percent or 500 ppm of Hoveda Grubbs 2, which is now called M72 Simes. This is at 0.2 molar concentration, you get quantitative yield. If you increase it to 1 molar, it still get quantitative yield. If you run this reaction neat, the yield starts to drop off, but it's actually not. If you're thinking oligomerization, that's actually not it. What it is actually is an isomerization product that can be suppressed by the addition of trifluoroacetic acid. So just a catalytic, I think it's 0.1%. I think twice as much, 2 to 1 to the catalyst of TFA is added, and that shuts down the isomerization reaction. So these conditions, these neat conditions were piloted, again, with including the TFA modification at 50 kilo scale. So it's certainly been proven out that this is a scalable way to make this reaction. And I'll note that it's run neat and just 500 ppm of catalyst. And I have a PMI calculation later, which I'll go through. Another nice thing that came out of that study is really a number of different examples where you can make either tri-substituted or tetra-substituted olefins by this. I've heard people say you can't make a tetra-substituted olefin by RCM. Yes, you can. You have to switch to this less hindered catalyst, so the orthotol instead of the mesotyl group on the NHT. But the yield is perfectly fine, and I would say this is not completely optimized. Again, this is coming from these are GC results coming from a 96-well screen, so it could be optimized even further. So of course, and as I said before, I talked about the azepane, there's a lot of different ways you can break apart a molecule, and that's certainly this is no exception. The more traditional way that you might think about breaking, and I think another challenge with RCM is that the way you disconnect a molecule is through a carbon-carbon bond. And that's, I would say with Suzuki couplings, maybe we've all learned to identify those type of disconnections, but I don't know that, I'm not sure that the RCM disconnection has really permeated the mentality, again, owing partly to the kind of business hurdles that had existed before. Of course, the more traditional way that you might disconnect this molecule is through the CN bonds, right? That's a polar reaction. The issue here, there's a couple of issues. This starting material, dichlorobutene, is a highly active alkylating agent. I think I'm not sure if it's GTI or potential GTI, but it's certainly something that you'd want to make sure is not present in anything that you would submit for human use. And actually, the other reagent, this is used as a surrogate for ammonia. This is hexamethylene tetramine. It's the condensation product of formaldehyde and ammonia. And of course, and this is used so that you get only monoalkylate, well, it's used in place of ammonia in this case. You do only get alkylation on one of the nitrogens. You don't get further alkylation. So it's only one of these nitrogens that's actually incorporated into the product. And the other problem is that this came from ammonia and formaldehyde you came, and from formaldehyde back to formaldehyde and ammonia you shall return. So those are all liberated in the course of this reaction. And also, making ammonium chloride and a number of other salts. These have to be washed away from your desired product, and that shows up in the PMI calculation. There's about 30 volumes of aqueous waste that's produced in this process. The last 2 steps are pretty straightforward. You freebase the amine, it closes rapidly, and then you put the Bock group on and you're there. If you contrast that with the RCM approach, so I think we actually purchased the NBOC diallylamine, but just for the sake of argument, I'll say we'll trace it all the way back to diallylamine, which I think everybody can agree is quite readily available. You can do the Bock protection, and actually this is where most of that 23 PMI comes from, is in this Bock protection, because that's kind of a traditional reaction in an organic solvent with a workup and everything. The RCM reaction, however, is run neat, 500 ppm of catalyst. You're only losing ethylene, so it's 28 grams per mole as a byproduct. So it's a very efficient reaction, low loading. So the PMI of that step on its own is pretty close to unity. It's about 1.1 something. So I think the combination of fewer steps and also definitely much more atom efficient route, I think that RCM certainly compares favorably to the more traditional method. So the last case study I'll talk about, and then actually I have a couple slides on optimizing these reactions. So another case study in process development, I think another prevailing notion, at least in the pharmaceutical industry, is that RCM is great as an enabling tool. It's something that the medicinal chemist will use to make something, and then, well, once it gets to process, we'll see if we can get rid of it. And that's true. I mean, there's many instances where that's probably going to happen. But there's also instances where RCM might even offer a better, shorter way to make a molecule in an nth generation route. And this is an example of exactly that happening. So this is Rolapton. It's an API that was approved in 2015. It was originally discovered and developed at Schering-Plough. It was divested as part of the acquisition by Merck. So this is an antiemetic, and Merck already had Emend on the market. So for regulatory reasons, they had to divest it. Schering-Plough had to divest it. It kind of was in the wilderness for a little while, was picked up by a number of different holding companies, eventually was developed and brought to market by a company called Tesaro, which was acquired last year by GSK. So the first generation route, both the medchem route and the first generation process route, went through, I would say, these are essentially the same disconnect. These are the disconnections that would occur to almost any organic chemist if they thought about how to disconnect this molecule. You might quibble about whether the etherification or that carbon-carbon bond forming step is a better one to do, but still. This is pretty much along these lines. In the forward sense, and here I'm skipping the medchem route, but even the first generation process route, the main issue here is that when you do install form this carbon-carbon bond, I actually didn't point these out, but this is a fairly small but very complex molecule. You've got two tetrasubstituted carbons flanking either side of this pyridine, which is part of a spirocycle. You've also got this chiral center, which certainly looks like a nice candidate for an asymmetric hydrogenation. So forming these stereocenters, this first one actually comes, so this is asymmetric hydrogenation. This one could come from phenylglycine, which it does. So this one comes from chiral pool. But in this sequence, at least the first generation sequence, the second tetrasubstituted stereocenter was formed in very poor selectivity. So here they install the nitro group to make a nucleophilic handle there. They do a Michael addition to an acrylate. And this is only described in a patent, so they don't actually give the ratio, but they do say that it's a poorly selective reaction. So that results in certainly a yield hit of the desired diastereomer at this stage. This is an improvement over the medchem route in that these can now be separated without any chromatography. They can be separated just by a crystallization. It still certainly left something to be desired to take that big of a yield hit. I don't know exactly how much, 40%, 50%, I don't know. So they went back, they thought about how else they might be able to disconnect this molecule. And they also noticed that this actually could lend itself to disconnection right through the middle of that piperidine ring, where this bond could be formed by reductive amination, and this bond could be formed by a ring closing metathesis reaction. And again, this bond down here is kind of the same as it was before. And of course, I'm skipping through lots and lots of steps, and also, again, this is only described in patents, so there's not really a lot of detail. But in the forward sense, they're coming back, tracing back to phenylglycine to get to this stereocenter. This one comes from asymmetric hydrogenation. Now the stereocenter on the top, the other tetrasubstituted stereocenter, also comes from a chiral pool material, in this case pyroglutamic acid. Under fairly standard reductive amination conditions, they bring these two pieces together using STAB. They form the tosic acid salt of the secondary amine, so there's not a protection group on, protection group off step. There's not an extra step added. They just need to salt this out. They do add a little bit of extra tosic acid to the ring closing metathesis step to make sure that basic amine stays tied up. They close that in good yield. I haven't shown the yield here. Again, and this is a similar theme as to before, these are the penultimate and the ultimate chemical steps are both transition metal catalyzed. However, I'd say that Merck example before, where you have less than 10 ppm of ruthenium and palladium after similar steps, maybe lends some credence that this might maybe be useful in scale as well. So with that, I'll kind of wrap up this kind of bond disconnection portion of my talk. And the main things I want to take away is that the questions about IP hopefully have been answered. Our model is simple. It's just you pay for the price of the catalyst and that's it. Grubbs catalysts have proven themselves in multiple different commercial applications, including API. And there are also, many potential applications other than macrocycle. And the other thing I'll say, this is the most pluggy thing I'll say, is that Umicore is here. We are chemists. There's many of us here that are all chemists, and there's more where we came from as well. We have the expertise to provide this kind of support. We'd love to be able to look at projects at early stages and make catalyst recommendations, additive recommendations, so on and so forth. And I'll talk a little, and I'll give us flavor of that in the next couple slides. So here's a quick user's guide. The first thing I want to do is draw attention to, here's the catalytic cycle. First thing I want to do, in case it's not on a slide, is draw attention to a really nice review paper from BMS earlier this year that kind of talks about a lot of the challenges that they've been able to overcome doing macrocyclizations using RCM. So I'm going to highlight just kind of a few things that stick out to me, things that oftentimes people don't think of, at least in my experience. They're surprises. So. The first thing I want to start with is just really. I told you there'd be no mechanism slides, but then here we go. So this is not a very detailed mechanism slide. In reality, these actually should all be reversible arrows to Neil's point earlier, because this entire thing is actually an equilibrium process. However, so what I want to point out is that what we sell you, what we put in a bottle that's a green powder or a red powder or whatever it is really a pre-catalyst, right? So once that initiates onto the substrate, starts turning over, it's really the propagating catalyst is determined by your substrate. So and in most cases it'd be 2-terminal olefins. If the substrate contains 2-terminal olefins, then the catalyst is really turning over, and the propagating cycle is the ruthenium methylidine catalyst, and that's an important point. So in terms of what's involved in those propagation steps, what do you still have on here? This is kind of I've stripped away some of the detail. What's still on there is the NHC ligand. So this certainly impacts the selectivity, also the stability of the propagating catalyst, as well as the rate of the propagating catalyst. The counterions can certainly play a role. They're still on there. But what's not involved in the propagation is this alkylidine, this lower portion, and nor is the second L-type ligand. So in fact, at least for the if there is a second L-type ligand, right? So if there's a phosphine on there, actually it's before the catalyst can initiate onto the substrate, that phosphine actually has to fall off. So that's not typically involved in the on-cycle catalysis. So in terms of the ligand impact, when I say this can certainly change the selectivity, this goes back to something I alluded to before, which is this OTOL catalyst, where a lot of the sterics of the mesothelium have been stripped back. I found this is a very stark example where the Hoveda-Grubbs2 catalyst that has the Cymez on it actually gives no reactivity at all to form this trisubstituted olefin, whereas switching to the cytol group, you see a dramatic improvement in yield, so it now goes to 80%, perfectly serviceable for medchem for sure. And with cross metathesis reactions, there's a similar theme when you talk about allylic branching. So if you've got a lot of steric bulk at the allylic position, then you also want to strip back some of the steric bulk that's on the NHC. However, this is a bit of a, not really conundrum, it's just a little more complicated. When you are trying to make a trisubstituted olefin by cross metathesis, you actually want a bulkier ligand, and this has to do with making the productive metallocyclobutane versus the unproductive metallocyclobutane, which I think is, well, I'll come back up anyway. So I guess suffice it to say, this is just a little bit of a flavor of, depending on what your substrate looks like, it may be certainly worthwhile to look at these other alternate NHC ligands, and these are catalysts that we do support, even including on commercial scale or development scale. So as I said before, the impact of the substrate is it really determines what the propagating catalyst is. And this is an example from more of the kind of the fine chemicals or specialty chemical space, where they're taking methyl oleate and doing an equilibration, basically. So they're getting both the kind of the homodimer that is all hydrocarbon. They're also getting the homodimer that is all has an ester on either side. But what they're doing is they're taking that equilibration is going to happen all the time, and it's always in equilibrium. It's going to be ever present. But if you react it, again, this is according to and I don't have the citation here, but the Chatterjee paper from 2003. If you expose it to an electron deficient olefin, now all of a sudden you'll now get a selective cross metathesis that gives you primarily these unsymmetrical products, and that was what these authors were after. And what they noticed is when they switched this cross-metathesis partner from methyl acrylate to methyl crotonate, they see a significant jump in both the conversion as well as the ratio of the cross-metathesis to the self-metathesis. And that ratio comes from the fact that, like I said, this is in constant equilibrium. This is going back and forth fast. These are also intermediates. Of course, this could go straight to react here, but so could either of these. And so when you switch to methyl crotonate, you now see good conversion across the board, even at very low loading. You go down to 29 ppm, and here you see mostly self-metathesis. Here at 29 ppm, you see a complete conversion and 97:3 ratio of the desired cross-metathesis product. And this is because you've changed your propagating catalyst from the methylidine to the ethylidine. And so now by just simply adding a methyl group here, you're shielding that ruthenium center from attack by nucleophiles, which is one of the primary mechanisms for catalyst decomposition. For instance, and this is work that was originally done in the Grubbs group, but also Darren Fogg has followed with some really nice studies on these decomposition pathways as well. So the resting state, so if the phosphine does come back on, so this is that they're propagating catalyst, except where phosphine has also come back on, so this is a resting state for the catalyst. If the cyclohexylphosphine is displaced by a coordinating solvent, that cyclohexylphosphine can then come back and attack at the, and again, you can think about this as it depends on whether you're an organic chemist or an inorganic chemist, I suppose. You think about this as a nucleophilic attack, you could also think about it as some sort of migratory insertion, but anyways, regardless, this results in a seized activation ultimately onto the NAC ligand, which is now you've gotten to, this is still a ruthenium complex, but it's certainly not an active metathesis catalyst. So what the study in 2016 by Darren Fogg showed is that if you basically switch your propagating catalyst from methylidine to ethylidine, you basically do not observe any of this decomposition product. Another common challenge that people have to overcome, especially again in pharmaceutical, very complex molecules, is you'll have basic coordinating groups that might interfere in the reaction. I showed before in the Rolapton example, you can just make the tosic acid salt. Here's another example of a macrocyclization. This was done at A-Star in Singapore in 2015, or at least published in 2015, comparing the activity of either M1A or M2A, that is Grubbs 1 or Grubbs 2, in the presence of HCl or TFA or versus not. And in the presence of the acid, these reactions are quite efficient. So having an acid there, again, it's not usually necessary to tie it up as a protecting group. Oftentimes you can try acid additives and that'll help. This is just a restatement of the previous conditions. Tosic acid salt, if that's convenient for your process to isolate it as a salt, you can just subject that to the conditions. And finally, the olefin isomerization, this is one that's very common, and these solutions have also been known for a while. And again, in that Merck example for vaniprevir, they used a different quinone, a very closely related quinone, with the two chloral groups on it as well. This can also be suppressed by the addition of acid. So the thought here is that there's a ruthenium hydride that's formed, and that's actually the isomerization catalyst. So that can either be trapped by just an acid or a weak oxidant. So, with that, I'll conclude my talk, and I'd like to thank you all for coming and making this symposium a success. It would also not be fun to have a bunch of talks in an empty room, so thank you all for being here. And I'd be happy to answer questions. Thanks.